Most of us walk around assuming our eyes work like passive video cameras, simply recording the world and projecting a raw, live feed to a viewing screen somewhere inside our heads. But modern neuroscience shows that this intuition is incorrect. If we look inside the brain, we won't find a central observer watching that screen, and there is no single region storing a complete, finished picture of reality. Instead, visual perception is a completely distributed process. The image of the world is broken down and scattered across millions of interacting cells. This diagram illustrates the core mechanism of hierarchical sensory processing. Sensory information passes through multiple sequential stages. At each step along the pathway, neural populations transform the data, recognizing increasingly complex patterns while preserving the relationships established at the stage before it. To understand exactly how this works, we need to follow a single beam of light as it enters the eye and track it deep into the brain. Tracing this pathway reveals that perception is an active structural assembly of information rather than a passive observation. The journey starts at the retina, located at the back of the eye. Far from being a passive sensor, the retina is a site of active computation. As soon as light hits the photoreceptors, subsequent layers, specifically the bipolar and ganglion cells, immediately begin analyzing and altering that incoming data. In this diagram, before the signal leaves the eye, the raw grid of light is processed to extract specific features. The retinal output emphasizes high contrast outlines and directional vectors to track motion, discarding raw visual noise. Those extracted vectors then travel to the lateral geniculate nucleus, or LGN, located deep in the thalamus. The LGN is a major processing center, where this incoming visual data is actively modulated by feedback loops returning from the cortex. Yet, throughout all of this data manipulation, the LGN strictly preserves the original topographical map of the outside world, ensuring that the physical-spatial relationships remain intact. So before the brain's cortex even gets involved, the visual data has been filtered and organized for action, rather than exact reproduction. From the LGN, the signals finally reach the primary visual cortex, commonly known as V1, at the very back of the brain. This visualization shows neurons in V1 do not process whole objects. They respond to elementary fragments, like specific edge orientations, isolated corners, or spatial frequencies. As the signal moves forward into secondary visual areas, V2, V3, and V4, the complexity scales up. Those isolated fragments snap together, generating enclosed shapes, solid surface textures, and 3D depth layers. Those elementary lines become actual surfaces, yet even at this stage, no single brain region creates the final object you experience. Instead, the object you perceive emerges step by step, driven by the rapid, successive integration of those simpler extracted parts. Visual reality is a bottom-up construction project. The complexity of the world we see is built from interacting fragments, never appearing all at once at any single stage. After this early cortical processing, the integrated visual data splits, traveling down two separate specialized pathways. The first is the ventral stream, which moves toward the temporal regions of the brain and acts as the what pathway. As data flows through the ventral stream, populations of neurons coordinate to build abstract representations, allowing us to recognize objects, to identify faces, and complex categories. Simultaneously, the second pathway, known as the dorsal stream, moves toward the parietal regions. This is the where or how pathway. The dorsal stream analyzes that same visual data to track movement and map spatial locations, specifically to guide actions like reaching and grasping an object. By splitting the data into these two streams, the brain allows us to identify what an object is, while simultaneously knowing how to interact with it. We can trace this entire pathway from beginning to end. Simple, isolated features build complex features, which build recognizable object representations, which then directly inform our physical behavior. The critical rule governing this hierarchy is that higher representations never replace the lower ones. The simple lines and the complex objects exist simultaneously, maintained through constant, coordinated interaction. Which leads us to the ultimate conclusion. At no point in the brain does a single neuron contain a whole object. The reality we experience is a distributed consensus across millions of individual cells. This hierarchical architecture demonstrates how the brain transforms raw input into structured neural patterns. These stable patterns serve as the biological basis for tokens, the internal symbols the brain uses to represent complex information. Understanding how the brain builds objects from fragments allows us to examine higher cognitive functions, like abstract thought or complex reasoning, emerge from these same principles of hierarchical integration. The brain does not take a photograph of the world. It builds it, layer by layer, in real time.